Hello, my name is Jaclyn Burke, and my presentation is on DNA detectives. I'm going to be talking about DNA technology, its biological foundations, and its powerful applications in forensic science, ancestry research, and more. Firstly, I'm going to be talking about the importance of DNA technology. DNA is the molecule that stores all the genetic instructions for life. Every cell in your body carries this code, and scientists have developed many technologies to study and use it. Some examples include cloning, gene therapy, DNA fingerprinting, and DNA sequencing. These tools are used in two major areas. One is forensic crime solving. DNA evidence can identify individuals with incredible accuracy. It has transformed criminal investigations by reducing errors, solving cold cases, and even freeing people who were wrongly convicted. Second is ancestry tracing. DNA testing helps people discover their heritage, trace family lines, and connect with relatives they never even knew existed. Now let's take a look at the biological processes that make DNA technology possible. The first process I'll talk about is DNA replication. DNA replication is the process where a cell makes an exact copy of its DNA before dividing. This ensures every new cell has the full set of instructions it needs. Here's how it works. First, the helicase unwinds the DNA double helix by breaking down hydrogen bonds between base pairs, forming two template strands. Then, the DNA polymerase adds new nucleotides to each template strand, building two identical DNA molecules, also while checking for mistakes. Then, the primase lays down RNA primers to start the process of replication. Lastly, ligase seals any gaps in the backbone. DNA replication keeps genetic information intact across generations of cells. This is essential for growth, repair, and reproduction. Without it, life couldn't continue from one cell generation to the next. Next, let's talk about the processes of transcription and translation. Transcription is when the DNA code is copied into RNA in the nucleus. Enzymes read the DNA and build a matching RNA strand, which carries the instructions to the ribosomes for protein production. Translation happens at the ribosomes, where the RNA code is read and amino acids are assembled into a protein. Each 3-base codon on the RNA matches a specific amino acid. These proteins determine traits and control body functions. That's why transcription and translation are so important. They explain how DNA becomes the physical traits we see and the biological processes we rely on. The last process I'll talk about is gene activation and deactivation, as well as gene expression. Genes aren't always active. They can be turned on or off depending on what the cell needs. When a gene is on, it produces proteins. When it's off, protein production stops. This regulation helps the body grow, repair itself, respond to stress, and conserve energy. Although not everyone expresses genes at the same level. Differences in gene expression explain variation in traits like eye color, hair color, and even susceptibility to diseases like cancer or cystic fibrosis. Gene regulation is also central to many DNA technologies used in medicine, agriculture, and forensics. Now I'll talk about how these processes and technologies show up in everyday life. My first example is forensics. DNA profiling is one of the most accurate ways to identify individuals in criminal investigations. It examines parts of DNA that vary widely from person to person, creating a unique genetic fingerprint. This has improved forensic science by reducing wrongful convictions and strengthening court evidence. DNA testing has also helped free people who were wrongly convicted. Groups like the Innocence Project have used it to overturn many cases, showing how DNA technology supports fairness and prevents major errors. An example case would be the Golden State Killer. In this case, Investigators use traditional DNA profiling and genealogy, uploading crime scene DNA to public genealogy databases, and building family trees to identify suspects through relatives. This method helps solve a decades-old case and shows how DNA technology can uncover leads that were once impossible to find. My next example is ancestry testing. Ancestry testing examines specific DNA markers passed down through generations to trace family history. It can reveal migration patterns and connections that written records may miss. DNA testing can also uncover details about your heritage and connect you with unknown relatives. This helps people rediscover family ties and learn about cultural roots, often giving a stronger sense of identity and belonging. Companies like 23andMe and Ancestry DNA use advanced sequencing and huge genetic databases to provide insights into ethnicity, family history, and even distant relatives, reshaping how we view our past. Lastly, DNA technology also plays a major role in health and agriculture. By analyzing specific genes, scientists can identify risk for diseases like cancer, diabetes, and heart disease. For example, BRCA testing can reveal inherited traits for breast and ovarian cancer, allowing for early prevention and personalized treatment. In agriculture, DNA testing helps breeders select plants and animals with desirable traits, like disease resistance or higher yields. This marker-assisted selection helps farming become more efficient and productive. DNA technology has transformed our world in several major ways. It has transformed criminal investigations by allowing precise suspect identification through unique DNA markers, helping solve cases faster and strengthening the reliability of court evidence. DNA testing also helps reunite families separated by war, disasters, or adoption, and identify missing persons. It often provides closure and hope when traditional records aren't enough. Another is DNA technology advances medicine and genetics, leading to breakthroughs like gene therapy and improving health through personalized treatments and a better understanding of inherited diseases. Despite its benefits, DNA technology raises ethical concerns like privacy risks and discrimination. These issues have sparked global debates about who should access genetic information and how to protect individual rights. These debates continue today. My question to those listening is, who do you think should control access to large genetic databases, and how should that access be regulated? Here are the sources I used. Thank you everyone for listening.